Hi, my name is Madison Bunch from the University of North Georgia, and my poster is investigating the question, do KT tapes increase skin exposure to disease-causing bacteria? Kinesiology tape, which I will refer to as KT tape, is a type of tape commonly used in athletics and physical therapy that is applied directly to the skin. KT tape was invented by a Japanese chiropractor, and they were released to the public in 1983. KT tapes are worn in hopes of reducing inflammation, preventing injuries, and increasing blood circulation in the applied area. Manufacturers recommend wearing the tapes for 3 to 5 consecutive days for the best results. We wanted to see if this prolonged contact between the tape and the skin will affect the bacteria that naturally lives on the human skin, known as the human skin microbiome. This skin microbiome aids in immune system development and provides a barrier to prevent infection. If the tapes affect the growth or abundance of the human skin microbiome, this could leave the skin vulnerable to infection. The aim of this study is to see if the prolonged wear of KT tapes affects the skin microbiome by causing an accumulation of environmental organisms, leaving the skin vulnerable to pathogenic bacteria. If this is the case, we also wanted to examine the antimicrobial resistance profile of these accumulated pathogenic bacteria. To study the effect of the tapes on the human skin microbiome, we evaluated 9 different types of KT tapes, one of which claiming to have antimicrobial properties. 10 volunteers wore all 9 tapes for 5 consecutive days, and the tapes were removed at the end of that 5-day period. Samples of the tapes were then plated on TSA agar and incubated for 24 hours. Following incubation, any bacteria that grew on the plates was isolated and identified based on their metabolic profile, using the biolog identification system. An example of a biolog plate can be seen at the bottom of the poster labeled as aerobe microplate. The identified bacteria were compared against published literature, and 10 of the identified species that were reported to be pathogenic were selected for antibiotic resistance testing. Antibiotic resistance was assessed using standard diffusion e-test assays with 10 different antibiotics, each from a different antibiotic class. Among the microorganisms isolated from the KT tapes, 21 bacterial species were identified. 3 were human-associated microorganisms, and 18 were primary environmental-associated microbes. Of this total 21 bacterial species, 10 of these were reported to be opportunistic pathogens. Table 1 shows the gram-positive bacteria identified, along with the tape they were isolated from, their reported origin, and any reported pathogenicity. Table 2 shows the same for the gram-negative bacteria that were isolated. Preliminary minimum inhibitory concentration, or MIC values, were obtained for 4 of the opportunistic pathogens identified. The values obtained indicated that each of the 4 opportunistic pathogens were sensitive to some antibiotics while being resistant to others. The sensitivity and resistance patterns matched expected patterns for some antibiotics whose activity is dependent on the gram type of the bacteria. Other MIC values that were resistant indicate the presence of antibiotic resistance genes and/or mutations granting drug resistance. In conclusion, continuous wear of KT tapes can affect the human skin microbiome by causing an accumulation of environmental microorganisms and opportunistic pathogens. This accumulation can leave the skin vulnerable to infection. The organisms accumulated also show varying degrees of antimicrobial resistance. These values indicate some drugs which are beyond clinical breakpoints, making treatment with those agents ill-advised. Future research for this project includes obtaining MIC values for the remaining isolated opportunistic pathogens. Thank you so much for listening, and if you have any questions, I will be attending the live Q&A session 5, Biology Session B, from 10 to 10:50 a.m. Thank you.